This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org. This is TWiM, This Week in Microbiology, episode 338, recorded on August 7th, 2025. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone. Where it's been raining cats and dogs, we are very soggy in Charleston for these past 2 weeks.
2 weeks, sorry about that, but we do need water.
We do need rain, yes.
Also joining us from Ann Arbor, Michigan, Michelle Swanson.
Hello, and it's lovely here in Michigan, kind of, in the low 80s.
Ooh.
Sun and blue skies. The smoke from the Canadian wildfires has finally moved on.
Our lovely neighbors. Also joining us from St. Louis, Missouri, Petra Levin.
Temporarily in Michigan, too, where it actually seems to be in the high 80s, but we're closer to the lake in Saugatuck,
nice.
Michigan,
Right,
and
Michigan.
It is very beautiful here.
You bet.
So can you see Petra, Michelle?
No.
I think she's about 2 hours away.
I'm
from
inland. Yeah.
That's very cool. All right. If you like these programs, we'd love to have your support. We give you science here on TWiM and all the other Microbe TV podcasts. And that's in contrast to a lot of what you hear in the press these days. So if you want science, no lies, no disinformation, listen to us. And if you can help support us, we'd love that. Go to microbe.tv/contribute. Before we get into our papers, I just wanted to point out here in New York City, we have a rather large outbreak of Legionnaires disease in Harlem.
Ooh.
Never good.
We have over 60 cases and 3 deaths. And I was doing a live stream with Daniel Griffin last night, and he was saying, what year was that Legionnaires convention in Philadelphia? How can you
76.
forget it? It's 1976, right? It's like
The
our
bicentennial. Yeah.
Exactly.
And the Legionnaires were celebrating, and they picked up Legionnaires disease. I guess it was from a cooling tower.
Yeah.
In the hotel.
Contaminating the air conditioning system in the hotel.
And that's likely where it's coming from in New York City. There are a lot of cooling towers for large buildings, as well as there are water towers affiliated with many of the apartment buildings in Harlem. And the last significant Legionnaires outbreak in New York City, they found it was associated with the water tanks on top of apartment buildings.
Yeah.
I actually have a question about Legionnaires, which maybe other people have, and maybe Michelle can answer, which is it was identified in 1976, but these water towers and these air coolers have been up above for a long time. Do people think that it had been happening, but just not a large enough outbreak to really identify the bacteria that caused it? Or is it really that it emerged in 1976?
It's a good question. I don't know of any data that addresses that.
Okay.
So nobody did serology to see if people had antibodies.
Yeah,
well,
but
the closest they have is Pontiac fever, in which there was a syndrome similar to how the Legionnaires presented in Philadelphia, and which is typically a pneumonia. But remember, Legionella is very challenging to grow. It requires special media, special conditions, and they just may not have had the technology or the media in order to elaborate the pneumonia. They knew that the patients had pneumonia, and that was Pontiac fever, which was in Pontiac, Michigan.
Yep.
Interesting.
Yeah, and it is more common in the summer because that's when the water in the cooling towers or people's use of whirlpools or hot tubs is higher. The water is warmer in the warm temperatures seasonally.
So
Yeah, that makes perfect sense.
hopefully this outbreak gets contained. Daniel was a little not pessimistic on how they will be able to contain it, but he thinks
Well,
it's. Go ahead.
because the Bronx had a really big outbreak a couple of years ago,
Yeah.
there is an excellent public health policies in place and expertise and training. So, for example, when the Bronx outbreaks occurred, they mandated that every cooling tower in Manhattan, or not Manhattan, New York Island, be remediated within like a 2-week period, which was just a heroic effort. And that, in fact, did knock down the cases. So I have confidence in the public health professionals there.
Yeah, especially since it's a state level thing
Yep. Yeah.
and not subject to
Yeah.
federal
But
issues.
just to put our listeners at ease, most people, healthy people that are not elderly, would clear this without any incident and not be aware that they were exposed. But if you're older than 50, if you've got some underlying immune weakness, or if you're a current smoker or former smoker, then you are more vulnerable to getting the infection, getting the disease.
Hmm.
So the actual incidence rate is actually quite low. I think only 10 to 20 cases per 100,000 people in New York historically get sick. So,
Yeah. All right, on to our papers. We have a snippet from Michelle.
yes, and it's actually aligns with this. It's about methods that we can more easily track nosocomial and also outbreak infections. The title is Automated Whole Genome Sequencing Platform for Bacterial Strain Typing in Clinical Microbiology Laboratories. It was published in the Journal of Clinical Microbiology in May of this year, and the authors are Rachel Siddell, Jordan Starkey, and Robin Patel. And they are at the Division of Clinical Microbiology and the Department of Lab Medicine and Pathology at the Mayo Clinic, and also the Division of Public Health, Infectious Disease, and Occupational Medicine at the Mayo Clinic in Rochester, Minnesota. So, what is bacterial strain typing and why is it important? When there is an outbreak, let's say of Legionnaires in Manhattan, it is important for the epidemiologist to understand where is it coming from. So you want to know that it's not just Legionella that is infecting the patients, but you need to know basically a fingerprint so that you can track back where is this Legionella coming from? Is it this cooling tower or is it that whirlpool, etc.? So classically, we used to use the antigens on the surface of the bacteria to serotype the different strains. And that was useful, but not all that discriminatory. Then in the 1980s, we started to get into molecular methods, and pulse field gel electrophoresis was used. So this method was you'd get genomic DNA and then cut it with a restriction enzyme that doesn't cut very frequently. So you generate these very large fragments of the genomic DNA and then run them through electrophoresis. But to get better resolution of these very large pieces of DNA, they would switch the direction of the current that was pulling the DNA sample through the agarose gel. And this gave more sensitivity for separating these very large fragments. The next advance was to use what's called multi-locus sequence typing. So the field would identify a handful of genes that are common to part of the core genome, and then they would sequence just those genes and compare the sequence. Then, most recently, we've been starting to apply next generation whole genome sequencing. So this is advantageous because it gives you much higher resolution, and you can also gain additional information, for example, about virulence genes, do they have them or not, and more importantly, what about antibiotic resistance genes? Because that would then inform treatment and help the physician decide how best to prescribe the right antibiotic. So the advance that we're going to talk about now is actually a comparison of a molecular method called core genome multi-locus sequence typing, which this group at the Mayo Clinic has been using to great effect. It is basically you choose a handful of genes that are part of the core genome, that is, they're conserved among many different strains of that species. sequence them and then compare the nucleotide sequence directly. But we're going to compare that method now to a new method that's been designed. And this is actually a protocol that's been developed by a company called Clear Labs in San Carlos, California. And Truth in Advertising, they are one of the funders of the research that I'll describe today. But it is a fully automated process that uses what we call next generation sequencing, whole genome sequencing. So I don't need to go into the details, but basically it's a way to efficiently get the entire sequence of the whole genome and then compare it bioinformatically. So you can really increase the sensitivity and discriminate whether a particular Legionella that came from a patient is the same as the other patient that died, 3 days ago. Or if you isolate Legionella from a cooling tower of building A, is it the same strain as what is in the patient or in building C? So is it one strain that is spreading, or is this just kind of background growth of Legionella, and we don't yet know where the spread is to the people who become ill? So those are some of the motivations. Does anybody else want to elaborate on that? Why having whole genome sequencing would be an advantage?
As you said, it's the fidelity. You can tell the subtle differences, and we've certainly experienced that as we're going through this evolution of SARS-CoV-2. We've seen the emergence, not necessarily of new strains of SARS-CoV-2, but we've just seen new variants that have a different flavor of spike. And you wouldn't necessarily see that in a bacterium that has a genome size on average of about 4 to 5 million base pairs to see a subtle difference like we're seeing in spike. And so the whole genome sequencing enables you to compare and contrast very quickly. People are probably quite familiar with the most famous of all E. coli's, E. coli 0157H7, the famous hamburger bacterium that they recall hamburgers with, and it
cause bloody diarrhea, which is unforgettable if you've had it.
And as Michelle pointed out in the beginning, the O stands for the O antigen on that microbe, and the H stands for the flagellum. And the numbers just mean there are at least 157 O antigens that we've identified in E. coli, and at least 7 different flavors of flagella. But at the same time, the sequence between E. coli that you use in your lab and E. coli that causes the Jack-in-the-Box syndrome is literally a remarkable, those genomes are actually different by about 60%. They only share 60% genes in common. So you can see, even though it's still E. coli, strains do indeed matter, and that whole genome sequencing will really give you a good understanding of this. A horse has actually now become a zebra.
Yeah, so the emergence of new variants that are pathogenic. Yeah. So not only tracking back to the source of the outbreak, but also is the pathogen itself evolving. So the Mayo Clinic has got deep experience using this core genome multi-locus sequence typing, but it's labor intensive. You've got to first isolate bacteria from the patient, prepare the DNA, then you've got to make a genomic library. And then after running the sequence, there's a lot of computational biology or computational analysis that has to happen to reassemble the genome and then compare it to databases. So they've found it to be very accurate and sensitive, but it's a lot of hands-on time and a long time to get the sequencing done and then analyzed. So what this Clear Labs has developed is a fully automated process. So they have a system where you start with 96 well plates. They then have liquid processors that can take the liquid and move it from vial to vial, step to step. They've got a built-in thermocycler for doing the DNA sequencing reaction, and then the reader, and then it's linked to the software that will assemble the sequence and give you the answer. So it's a lot less hands-on time, less manual labor, therefore less risk of like repetitive stress injury from all that pipetting that must be done, and also less opportunity for human error. So that was their motivation, the lab's motivation for testing this new automated system and compare it to their tried and true. And this was really a large effort. They analyzed 226 different bacterial clinical isolates that came from multiple collections across the country, and one set came from France. They represented 18 different species, and these are shown in Table 1, and there are many of the pathogens that you will have heard of, infamous, such as Legionella, which we just talked about, E. coli, which we just mentioned, also Staph aureus, et cetera, et cetera. So this was really a broad look to directly compare these 226 isolates analyzed by their core genome multi-locus sequence typing versus this automated process. So what they've done is compared multiple parameters. Importantly, of course, they wanted to calculate what's the total turnaround time. That is, from collecting a patient sample until having the answer. What is it? What are the particular allelic changes or similarities, et cetera. They also quantified the number of hours of manual labor required, the number of hours of processing time. That is, you don't need a technician there, but the machines are determining the sequence, and then doing all the alignments and comparison. And then importantly, they also looked at the costs. So all of those are important. Of course, the most important is you not give up on the accuracy, the fidelity of it. So they present their data in lots of detail. So for example, Table 2 shows us the average time per step for manual and automated whole genome sequencing methods. And we see that with the manual whole genome sequence method, it takes about 5 hours of hands-on time, whereas this automated system only takes 2 hours. And then the total time they calculated was, with their current method, it takes about 44 to 47 hours for running one set of samples. And that has been reduced now with this automated method to 28 hours. So you're saving almost a day. And if you've got a patient that is not responding to your antibiotic, for example, or you don't know what they're infected with, or you don't know whether this is something that is spreading through your hospital or spreading in a neighborhood in the Bronx or Harlem, for example, saving a whole day is going to make a big difference in healthcare. They also, as I mentioned, looked at the cost, and that is detailed in Table 3, where they know what it costs to do, to have the technicians, the skilled, trained technicians, what it costs to train them, the materials that are required, the salaries of the laboratory directors that have to oversee this process, the overhead, et cetera. And they compared that to this automated system. And they found that they can reduce the cost by around 34 to 48, 49%, depending on how many samples they're running in each batch. So you don't just run this whole sequencing system with just one sample. Ideally, you wait and have multiple patient samples and then run it all through and get the answer. So with this new system, I think they can run up to 12 simultaneously, and they're getting significant cost reduction. So this is pretty exciting. They think that they'll more quickly be able to come up with a treatment plan, and also begin to look at infection control procedures if they're discover that there's a outbreak spread in their hospital, for example. And then, of course, saving strain on their colleagues' arms and wrists from all the manual pipetting, and overall reducing the healthcare cost. So this is exciting, and there are many applications that could be done, not only in a clinic, but also epidemiological investigations, such as the outbreak of Legionnaires disease that we just talked about in Harlem, in New York.
Michelle, this is really amazing, because every time I go to like the ASM big meeting, there's always all these companies trying to like figure out, you have to crack this code, because not only for understanding if there's resistance or what the organism is, but also for stewardship. So you're not always giving, for example, ciprofloxacin, which is a broad spectrum. So how close is this to being something we can implement? Because right now sequencing has been behind and people have been trying to use mass spec, which obviously doesn't pick up some of these.
Well, I can say that this is this system from Clear Labs has actually been on the market and available. It was previously applied in Milwaukee during the SARS-CoV-2 epidemic, and they used it for surveillance and tracking variants. It's also been used for wastewater surveillance during the SARS-CoV-2 epidemic, and they've also used it for mycobacterial species surveillance and also looking at antibiotic resistance. So the Mayo Clinic, I believe, was the first to do this really broad test of 226 different clinical samples. And importantly, they found that the fidelity between their traditional current system and this new automated system was greater than 99%. So they're not giving up anything in accuracy, but they are gaining cost savings and also speed.
Wow.
And with each passing day, we're getting better and better bioinformatics, principally because of the revolution in the graphics chips that are used to do the assembly. And as companies see investment by healthcare systems, researchers in this technology, they'll be more likely to add the newest and best chip, which will then go to reduce the time to you get your piece of data. And, in the olden days, we didn't have the CPU power that we have today. And with each passing day, we see the revolution in these graphics chips that are involved in the assembly of the sequences from this unbiased shotgun sequence analysis. So where I see this really going next is you can imagine such a system being used to ask a question, did the probiotic I take? Did it actually establish itself in my gut? Because how else will you know if the strain that you want to make more butyric acid in your microbiome is actually still there without going through heroic work? And so it'll be the strain analysis that will really drive the adoption. And I think that's where, aside from the clinical space, this is where this technology will really see widespread adoption as we begin to do more and more clinical trials for the microbiome and probiotic world.
Mm-hmm.
Michelle,
It'll advance our research then as well.
Absolutely.
Like the example you put out. Yeah, Vincent, what's your question?
so this study was done with isolates, right?
Yes.
Can you use clinical samples, or do you need to have an isolate first?
I believe they were isolated first.
Okay.
So you take the sputum and isolate the bacterium. Yeah. So then they started the timing and comparison once they had the culture.
Because I think we've done a paper before where they take specimens with the blood. I think the paper we did was on blood,
Mm-hmm.
and just try and do whole genome sequencing on blood, because that's obviously faster than having to get an isolate, right?
Yeah, although the computation would be a bigger
right.
time sink.
Yeah, I mean, I think that's the gold standard, right? If we could get it directly and amplify it and figure out what the bacteria is that's causing the problem and whether it has drug resistance markers. I think that's why people have been using mass spec, is my understanding, even though mass spec doesn't tell you about the resistance, it can give certain signatures for the organism.
Right.
Because it's a data culture. So basically you're at the same point you would be in terms of understanding the organism as you would, because once you do that in the micro lab, the sequencing is only going to tell you what the other markers are, right?
Right.
You should know by culture what it is in 24 hours or less.
And you have to be very careful that the isolate that you begin with is indeed a pure culture. And so you have to strict isolation, make sure that it doesn't have a lot of contaminating microcolonies within it. So you have to streak it 4 times before you're really convinced that it's breeding true. And the other limitation that I was worried about when I read this paper for the first time is about DNA contamination, because we're always taught that you grow the bacteria in a different place from where you're doing the DNA isolation, and that DNA isolation you want to occur in a clean room. And this technology sort of takes care of that by its microfluidics and its isolation of the equipment. So it gives you a better appreciation that cross contamination may not be as common as it was in the manual way. Because if you've ever been to a clinical lab, you know there are literally thousands of Petri plates growing microbes. There's DNA everywhere. And it's truly a challenge to get clean DNA. And where they do the DNA isolation is also often in a separate, down the hall, away from everything room.
Yeah, and the authors make that point that this
Yes.
is all contained one place, so they're not having to do what they currently do, which is isolate it one room, then take it to another place for the whole genome sequencing, the library prep, et cetera. They do point out that they have a relatively small sample size, so it will be important to verify their high fidelity that they obtained with this method using additional species, and also they recommend further testing with isolates from additional outbreaks to really test the sensitivity. But they're enthusiastic at this point.
All right. Thank you, Michelle.
Yeah, and thank you to the people at Mayo Clinic advancing epidemiology, patient care, and infection control.
All right. Now we are going to rewrite the code of life with Petra.
Ah,
Okay, so I saw this paper first because it's there's a very nice article in the New York Times that goes with it, I believe by Carl Zimmer. But this paper is entitled Escherichia coli with a 57 codon genetic code. It's by Wesley Robertson, Fabian Rem, Martin Spink, Raphael Schumann, Rongjin Tian, Wen Liu, Yangji Gu, Oscar Kleefeld, Cicely Day, Kim Liu, Yanka Kristova, Jerome Zurcher, Frank Bouge, Jaco Birnbaum, Linda van Bijsterveld, and Jason Chin. And they are all at the Francis Crick Medical Research Council lab in Cambridge, UK. And this paper is actually kind of a paper on a series of papers, like a process where people have been trying over probably a decade to reduce the genome size of, not the genome size, but the genetic code. So I think most of us learn in biology class that there are 64 codons. You get those nice little charts, right? There are 4 bases, A, C, G, and U for coding for amino acids. And those can be arranged kind of in a 64 grid. And 61 of those different, a grid of 3 letters, A, C, G, or U, are dedicated to an amino acid right? You have a tRNA and an anticodon and a charged tRNA with an amino acid. And you need 3 letters to code for an amino acid. So there are 20 essential amino acids, and you get 61 codons, these 3-letter codes for amino acids. So there's a lot of what we call redundancy, where you would have 3 letters code for, you might have, I think for serine, there are 4 different codons for serine, 4 different 3-letter codes for serine, et cetera. So there's a lot of redundancy in there. And then there's 3, so 61 codons for amino acids and 3 for stop codons, because you need to code for the ribosome to stop making the protein or you would never end. So the goal has been to essentially get rid of some of the redundancy. Instead of having 4 different codons, these little 3-letter codes for amino acids for serine, for example, maybe you only have 2. And why would you want to do this? I think is a really good question, because this is actually difficult to do. And I think the biggest reason would be because, for example, this is all done in E. coli. We use E. coli to make proteins that are really important. For example, insulin is made in recombinant E. coli. And maybe you might want to put an amino acid that we would call unnatural, maybe a variation of an amino acid that occurs in nature that might make a drug more effective or might make it an organism resistant to a viral infection. And so if you can cut down, essentially free up maybe 5 or 6 codons, these little 3-letter amino acid codes, to use them for different amino acids or different molecules that look like amino acids, then you can kind of change what the content of the protein that's being made by E. coli and maybe use it for a different purpose. So that's the premise and why you might spend a lot of time trying to take 61 codons down to 57 codons. And again, there's a good amount of redundancy. Most amino acids have 2 or 3, or like I said, even 4 3-letter codes for them. So one of the other questions is why all this redundancy? And one reason is actually that even though serine has 4 different codons that work in E. coli that will all give you serine if you see them in a genetic sequence, E. coli really only prefers to use maybe 2 of those. So it has tRNAs that tends to make tRNAs that only recognize 2 of those codons, and the others are rare. So most times when you see serine in an amino acid sequence in an open reading frame for an E. coli protein, it will probably be one of 2, maybe 3 potential codons. So just saying, well, why don't we get rid of all the redundancy? It's not very easy. The other problem that they have is that often coding regions in bacteria can overlap. And so if you change a codon in one protein, in one open reading frame, that's going to change the sequence of that open reading frame. And that might impact the other overlapping reading frames. So that's another problem. And then, of course, whenever you change DNA sequence, you alter RNA sequence, and that can alter the secondary structure of the RNA because RNA is single-stranded and often folds back on itself. So first, because of codon usage, that's a problem because you want to make sure the codons you're eliminating are not ones that are common for E. coli. You also want to make sure you don't interfere with genes that overlap or maybe are transcribed on the bottom strand. And you also want to make sure that you don't alter the mRNA structure, right? So that you change the folding properties of the mRNA. So with all this in mind, this group decided to try and reduce. They'd done it before, reducing, I think, by 2 amino acids. Now they want to reduce it even further. And so they use this is actually a really nice paper to read because it's very structured from top to bottom, like how they do it. The first thing that they do, and I thought this was very smart, is instead of trying to recode the entire 4 million base pair E. coli genome, they pick 20,000 bases to try and recode. So they pick genes that are really important or essential in E. coli. They found a region of 20 kb or kilobases of 20,000 bases that encode a bunch of important genes. And then they tried different schemes, like which codons can they change without having too much effect? Can we make serine, these 2 versus these 2? And they try different schemes, and they find actually several that seem to work in their sample size. And so they just basically pick one of those and then they scale up. So again, you're going to try and make a synthetic E. coli in which you essentially recoded the entire genome, but now instead of having 61 different codons for amino acids, you have only 57 codons for amino acids. And so how do you do that, right? You're not going to synthesize 4 million base pairs and then, I guess, just hope that somehow you get an E. coli out of it. You have to do it in a stepwise fashion. So they basically synthesized shorter fragments and then assembled them in yeast, in Saccharomyces cerevisiae, by recombining them into something called a bac, which I really think it's kind of like a giant plasmid, a giant circular piece of DNA. And because yeast are so good at recombination and it's so stable, they can take up to 100 kilobases that they've recoded with the reduced number of codons, and they can essentially put it into the bac. So now they have a yeast strain that has a giant circular piece of DNA that encodes 100 kilobases, 100,000 bases of E. coli DNA. And they have about 40 of these strains so that they all match to the 4 million bases of E. coli's genome. So now they have a bunch of yeast with giant circular pieces, and they have to put them into E. coli. So is that clear for everybody? Sorry, there are a bunch of steps here.
It is, but they were.
Okay.
It becomes obvious when they describe their troubleshooting why they were so systematic, because sometimes the changes they made had a real significant effect on growth of the E. coli. So then they could focus on that 100 kb element and troubleshoot just that instead of the whole 4 million base pairs.
Exactly. They really break it down into pieces. So now they have 40 yeast strains with 100 kilobases. So the first thing they have to do is get 100 kilobases, 100,000 base pair stretch of DNA into E. coli using these facts. So these giant circles. So they can prepare them, and then they use a CRISPR modification, which is actually figure 1 supplemental, which I suggest people look at because it's very clever, because they have a circular piece of DNA. It's very large. And anyone who's transformed E. coli, it can be pretty picky, especially the bigger the plasmid. And this is massive. So they need to get it into E. coli. Then they use this CRISPR method, which, right, CRISPR is something that can make double strand breaks. So they have special signals that the CRISPR recognizes. So it clips the giant circular back piece of DNA and linearizes it. And then they use something that's been around a very long time from Barry Wanner's lab that almost every E. coli lab, including my own, use all the time, called lambda red, where they get recombination, where essentially they have a recombination gene that they can recombine it into the genome. So essentially they do 100 base pairs one at a time, E. coli strain after E. coli strain, to get this to work. And as Michelle said, there are problems, and they knew there were going to be problems because of reasons I told you, right? Some codons are rare, overlapping genes, mRNA folding, all sorts of things. And then when they have problems, and they actually most of them, they seem to be able to get through, but they have some of these 100 base pairs that don't go in very well at all. And they can know that. And the way that they know that is when E. coli is unhappy, it grows really poorly. And so this is a way you kind of measure fitness in E. coli, is how fast it grows. So if it can't make important genes well, because it has now these recoded codons, it grows really slowly. And they actually generate a whole fitness map where their recoded regions don't work. And they even have some that are so bad E. coli can't deal with them at all. And they can't get them into E. coli at all, these giant backs. They can't get the recombination, or they get smaller regions where they can't get recombination. So essentially they go 100 base pairs, put it into E. coli, see if the E. coli grow at all, see what the rearrangements are by sequencing, and then try to narrow down by essentially looking at sequence where the problem regions are. And so by doing that, they can actually identify smaller regions where they have problems. These turn out often to be in regions of regulation, possibly because of folding. And then they go in and they try their different ways of getting to the amino acid sequence they need. One of the things that actually I thought was kind of satisfying, although I can't imagine doing these experiments, they're very slow and tedious and kind of frustrating, is that they find the most problems are near the N terminus, right, where the protein is being synthesized. I think this is probably for 2 reasons. Again, if the mRNA is changed in some way or folds differently, that can actually make the ribosome fall off. But also, if the codon doesn't have a matching tRNA, or there are very few of them, that's going to slow down translation. And that can also lead to a block in translation. And so for those 2 reasons, they focus most of their attention on recoding in the N-terminal regions of the open reading frames, or the ORFs. And that they can, and again, they're just going one by one. So they figure out where all the problems are, and then they tackle them systematically one by one to essentially fix the problems.
Yeah, I agree. This must have taken a lot of patience, but yet they know that their long-term goal has such great potential and applications that they know it's worth the hard work.
And they'd be doomed if they didn't do this systematically.
Right.
Mhm.
Right, because if they just let evolution essentially fix the problem, then they wouldn't necessarily end up with the codons that they want.
I mean,
So,
that begs the question, why has Mother Nature, since Mother Nature always wants to streamline the amount of baggage it carries to the next generation, why does Mother Nature perpetuate the 6-box amino acids, the 4-box amino acids?
For reasons we don't know, Michael,
Yes,
because they would be gone if we didn't need them, right?
yes.
It has to be now. Even though this E. coli can grow this way, I bet in nature it would be tough.
Well,
right. And codon usage is just amazing to me because in theory, ACGU are essentially present equal amounts.
Hmm.
Why does E. coli have a slightly different codon usage than other bacteria and different from human codon usage? Why are there these codon usage preferences? Are they just something that's been selected for? Are they somehow advantageous? Or is that just kind of where evolution ended up? And they're, you can imagine the cells encode all of the potential tRNAs, but they only use really a couple. So it could just be some kind of spontaneous like selection, but it's really unclear. And so I think this is a really interesting problem. But in terms of this application where you want to be able to free up genetic space to put in artificial amino
Hmm.
acids
Hmm.
or other modifications, it's actually very challenging because each organism is super adapted to its own particular codon usage preferences. So they managed to get essentially 39 of these all done. And again, these are going one by one. So they're creating 39 different E. coli strains from 39 different yeast strains with these 100 base pair recoded regions. So essentially you have 4 megabase genome, but only 100 is actually what it is. And then they start putting them together again systematically into individual strains. So they start to take those 39 strains and condense them down because, again, their ultimate goal is to have one strain that only uses 57 codons. And they, again, do this systematically. And then they essentially are using conjugation. So now they are using a way of transferring DNA between two organisms using this conjugative pilus mechanism to transfer DNA. And so again, they go systematically one by one, creating a larger and larger fraction of the genome in the new engineered strains that is recoded, that is synthetic.
And to think about it is if you've ever puzzled your way through conjugation, looking at the difference between an F plus strain of E. coli and then a HFR variant of E. coli. This is why conjugation works. You literally drive the new piece of DNA into the chromosome of the recipient and you force it in there so that these strains can effectively conjugate and you're going to build the chromosome as you build it up in a systematic way. You take the first one, you conjugate it in, you get a stable structure. Then you take the next one, you allow for the recombination event. So now you have, if you will, H prime, HFR number 2, and then HFR number 3. And it's remarkable what they did.
Right. So they're doing 100 kb, then they get to 200, 300. And along the way, they have problems, right? Because now sometimes some of the strains, none of these strains, even when they try to optimize the recoded region, some of them aren't that fit. They don't grow that well and they don't conjugate very well. So sometimes they have to essentially select for isolates of the donor strain that actually are better at conjugating and do it again. So each step along the way, they have to like systematically, like we've got this far, we have to get past this roadblock, and then they'll have another roadblock and another, and they just systematically go through. And they're always checking to make sure the genomes are stable and the sequences they want. So again, this is a super heroic effort. And in the end, they generate this strain that they call SYN57. which is essentially all synthetic genome that has 57 and doesn't need these extra tRNAs. They presumably have gotten rid of the sequences that even code for tRNAs they're not going to use. So now they have this 57 strain. And even this strain, which they've worked really hard to get to, has a fitness defect. It grows about 4 times more slowly than wild type E. coli. So wild type E. coli lab strain grows probably in the rich medium. They're saying 2xyt. So that's very rich medium. It's full of things E. coli likes to eat. And that E. coli usually doubles every about 20 minutes in 2xyt medium. Their strain doubles about every 80 minutes. So it's growing 4 times more slowly. But that's still pretty good considering they've gotten rid of 4 codons. So it shows like, even though there is all this redundancy in the genome, the bacteria really, really care about it. And it is not trivial to do this. So I think really the message of this paper is you can manipulate a genome, but it actually is difficult and takes a lot of time to do. So this kind of fantasy, when I first saw this, I'm like, oh, did they just synthesize a new genome and put it in essentially a membrane sac, an anucleate cell, and then that propagated? No, not at all. You can't do that. That seems to be the way to do it in my head. But in reality, you have to go the long route, as you often do in science, and kind of systematically do step A, step B, step C, step D. They also did all this beautiful characterization, but now they actually have a bacterial strain where they could essentially add up to 4 synthetic amino acids if you wanted to make a protein with specific chemical handles on it or markers or something like that. You can now do that by feeding E. coli those amino acids, having a tRNA that essentially can stick them on its end, and you could add them to the E. coli pretty easily to the SYN57.
So great.
An 80-minute doubling time seems slow, but Saccharomyces cerevisiae has 120-minute mass doubling time. Slow for bacteria is still pretty fast.
So obviously there's a fitness cost of doing this, right?
Oh, yeah.
Yeah. But the potential for generating new compounds that could be useful in research or in medicine is really exciting, or engineering of other types.
I don't know if you guys remember, but on TWiM 330, we talked about making E. coli with just 1 stop codon.
Mm-hmm.
Yes, I think that was a step towards this, was where they got rid of, there are now 3 stops, and now then you can go down to 1 that gets rid of 2 codons.
Yeah.
So, so then you're down to 62, and then I think they took it down to 60. Yeah, so essentially, methodically, in theory, they could take this strain, because there's still a lot of redundancy in terms of codons to amino acids. There's still a lot more multiple codons for specific amino acids. So in theory, you could actually keep taking it down.
And these authors really did heroic work, because when Mother Nature wanted to add another amino acid, selenocysteine, she used a stop codon, the opal codon, and she engineered a selenocysteine element within the RNA. But the bio on these authors is actually quite remarkable. Wes carried out his PhD in chemical biology at Yale University with Professor Alana Scherparts, where he gained his initial interest in genetic code expansion. So as a postdoctoral scientist, he jumped across the pond, as they say, to join the lab of Professor Jason Chen at the MRC of molecular biology in Cambridge. And Jason also performed his PhD research in Alana Scherparts' lab at Yale. So it's sort of the grandson is going back to the son who left home, and now they're continuing on, or the older brother, I guess, if I'm doing my genealogy right. At the LMB, he worked on re-engineering the genetic code of E. coli to enable emergent applications in the emerging field of synthetic biology, such as viral resistance and unnatural polymer synthesis. And now he started his own lab at the LMB, and last summer in 2024, where he pursues synthetic genome research to advance the fields of bacterial genome design and total genome synthesis, with the ultimate aim of bringing the benefits of synthetic biology to the field of microbiome research. One of his strongest, especially interesting or exciting days in the laboratory, and as you might imagine, for the assembly of the SYN57 genome, the final conjugation step was performed in late April of 2025. And this final step was to integrate that last 64 kilobases of recoded DNA into the fully synthetic 4 million base genome and analyze it via NGS. And that was the exciting experience with this team of co-first authors. And that should be emphasized. There were a lot of first authors on this paper, given the heroic effort that all of these workers had to put forth. Once they had the NGS confirmation, they had a fully recoded genome. They knew it was time to celebrate. And so, like good Brits, they made sure they properly capped off a successful day with a well-deserved trip to the local pub, inviting other members of the lab and, of course, Jason as well.
I want to go to the pub now.
Yes.
Congratulations to you all.
That's
It's
great.
amazing.
Yep, a real tour de force. Really great.
So Ted also has advice for junior colleagues or aspiring scientists. My main advice for aspiring scientists is to find which topic motivates you the most. What motivates you to continue to ask questions, to continue to learn novel techniques, to answer your questions, and to continue to work with others until you've answered those questions.
Yeah, otherwise it's just work.
Mm-hmm.
Yeah.
Well, I think that's what this paper really illustrates well, is this would not have been possible without a collaborative environment.
Right.
This is not trivial tasks where you can knock it out in a month. I mean, they did a lot of work.
Yep.
Yeah.
Yep.
This is definitely a big team effort to
Sure.
get through this.
For sure. All right. That is TWiM 338 here in first week of August. Summer's coming to a close, folks, so enjoy it.
Yeah.
You can find the show notes at microbe.tv/TWiM. If you enjoy these programs, we'd love to have your financial support to continue them. Go to microbe.tv/contribute. Michelle Swanson is at the University of Michigan. Thank you, Michelle.
My pleasure. Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levin's at Washington University. Thank you, Petra.
Thank you so much.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.